So originally we used to do this test in the other lab, but we switched to doing it in this lab because, as you guys already experienced, that lab was a lot, and we still didn't even do some of the stuff we normally did. Remember, we did the inoculations the night before, so we probably still have some moving around to do to try and make it so that some of these labs aren't so packed. So flavoprotein, what is that? It's an electron carrier in that electron transport chain, and it's one of the ones that transports those electrons to oxygen. Remember, this is for aerobic organisms like ourselves, and most of your organisms can respire aerobically, right? In fact, all of your organisms can respire aerobically. Some of them can only respire aerobically, where others are facultative anaerobes and can actually ferment when there's no oxygen. They don't have to use their electron transport chain. So they've all got all your organisms have electron transport chains. They all have flavoprotein. They're all, if oxygen is available, transferring electrons to that oxygen. The problem is that, remember, sometimes they jump ship of the electron transport chain before the end, and they form these toxic forms of oxygen. So to help protect us from those toxic forms of oxygen, we have enzymes. Superoxide demutase converts that superoxide radical of oxygen into safe oxygen and hydrogen peroxide. And then catalase, which is what we're going to test for the presence of today, can break down that hydrogen peroxide into safe gaseous oxygen in water. And so this is why, right, and they're both redox reactions, right? Somebody's losing electrons, someone else is gaining them. The electrons are moving. So catalase is the enzyme that's converting the hydrogen peroxide into gaseous oxygen, and that's why it bubbles, right? That's why hydrogen peroxide can bubble. So we're going to do two tests. We're going to do the slide method test, and I have some sterile plastic loops out so we don't have to mess with Bunsen burners tonight. And we can put the bacteria on the slide. And also, if you were using the metal loops, this procedure has to be done in the correct order, right? You have to make sure that you put the bacteria on the slide and then add the hydrogen peroxide. But if you're using plastic loops, you don't have to worry about that problem of cross-reactivity with the metal loop in the hydrogen peroxide. So that's another thing that'll make it really easy for us. So you're going to put your organism on the slide, and we're going to add some hydrogen peroxide to it. You should hopefully see bubbles, right? Because as I said, all your organisms have an electron transport chain, right? That can use oxygen as the final electron acceptor. So everybody should see bubbling. The one organism that we had that didn't bubble was yet again a pathogen. So I have to find us a new non-bubbler. That I need to find at least two, because, you know, with that one test, you know, you would figure out what organism you have. But some of our other tests are like that too. Takes the fun out of it. So we're going to actually put it right on the slide, right on the slant, excuse me, after we do the slide test. You do have a slant. You did label catalase, but it's quite old, right? And especially for those of you guys that know you have spore formers, that might create a problem. You might not actually see any bubbling. So you'll notice that you have a fresh culture that I made for you guys yesterday if your organism was grown at 30 degrees, which are the two back tables. You'll see that there's one that was dated yesterday with a little circle and your number. And you can use that one, and I would suggest using that one definitely for your slide test, and you can even use it for your slant test. I'm going to do a control, a known positive, and I'm also going to do the uninoculated control. And this will happen within seconds, right? You'll see those oxygen bubbles forming. So what would a false positive for the reaction between the inoculating loop, if you were using a metal loop that is, and hydrogen peroxide? Is that caused by poor specificity of this test or poor sensitivity? It's a specificity issue, right? You don't know whether it's the organism catalyzing the reaction with catalase or the metal loop causing that reaction to happen. So you've got to do that procedure in the correct order, or you can kind of cheat like we're doing tonight and just use a plastic loop and eliminate the metal problem, right? And this is a cool YouTube video where the guy exposes silver jewelry, and it's actually silver is one of the metals too that'll catalyze that reaction. So it's a specificity issue. So we can avoid that, right, by either doing the procedure correctly or using metal loops. Plastic. If you have a weakly catalase positive, say you put your organism on the slide, you add the hydrogen peroxide, and you can't see the bubbles, and you don't look at it under the microscope to help you see those bubbles, you might record that as negative, right? Would that be a false negative due to poor sensitivity or specificity? In this case, it would be a sensitivity issue, right? You can't see it. Why is it advisable to perform the test on a known catalase-positive organism along with the organism you are checking? Well, this is a control, right? What could go wrong here? So this brings us to our reagent. It's hydrogen peroxide, right? Why do we keep it in a dark bottle? Light sensitive. Light can actually cause it to turn into water and oxygen. You ever open a bottle, it's been closed for a while and it kind of fizzes? That's the oxygen gas releasing. Chances are that's probably a pretty old bottle in that case, and you might want to just throw it out because it's mostly water at this point. That gas has released from its liquid form. There's also an old wives' tale, if you pour hydrogen peroxide on a wound and it doesn't bubble, right? It's not infected. No, you just poured water on yourself. It should always bubble because, remember, you are catalase positive, right? Your cells should break it down, whether the bacteria are there or not. So we want to make sure our reagent is fresh. So actually the stuff I had the other day was old, and so I dumped it and got new stuff. So we get nice fresh stuff. It's already been tested. It's going to bubble like crazy. And this stuff's cheap, right? You can buy it for like 80 cents at Walgreens. It's not a big deal. They don't advise that you use it to clean wounds anymore, though, because it causes too much destruction to your own cells. So good old soap and water and triple antibiotic ointment. Neosporin if you're rich. Yeah. I buy the cheap stuff. It's the same damn thing. Why pay the extra $4? Okay, so what's the purpose? And I'm going to do this. I'm going to add hydrogen peroxide to an uninoculated tube. So I have a tube, a slant tube there that doesn't have any bacteria in it. And I'm going to add hydrogen peroxide to it. Should anything happen? No. If it was to bubble, could we do this test? No, because you wouldn't know if it's the media that's causing it to bubble or your organism. So if the media had the ability to break down this and cause it to bubble, then we couldn't do the slant test. But we can, but we're going to be sure that the media that we're using, which is Nutrient Agar, does not cause the hydrogen peroxide to break down and bubble. So all about controls, right? In this case, a negative. All right. So this is important. These scientific methods, right, although I've been kind of breezing through them, these you might want to go back and look over because it's an excellent summary of everything we've done, right? So why are we doing this test? Well, we want to know, right, does your organism have catalase? Does it have an electron transport chain where it can transfer electrons to oxygen, right? So this links to another test we performed, doesn't it? What test told us about oxygen requirements for our organisms? The FTM, right? Some of you guys, your organism, that's the only choice, right, is aerobic respiration. For some of you guys, it can ferment. It did grow in the bottom of your tube. It is a facultative anaerobe. So again, how do you perform the test, right? And make sure that you perform it correctly. What are the outcomes? Well, if you see visible bubbles, you know that oxygen gas is being produced, that your organism does have the enzyme catalase. If you don't, right, remember, sensitivity can be an issue, right? We would want to check that under the microscope. What can we say about our organisms? Well, the organism that exhibits catalase activity surely have an electron transport chain, right, that uses oxygen as its terminal electron acceptor. They respire aerobically, which is the case for all of your guys' organisms, right? So those little write-ups at the bottom, you have it. The answer was on the past slide. I just looked at it. Okay, so the next one is a citrate. All right, so this is just an interpretation of the results. And so in the blue racks with my name on it, you can see what a positive result looks like and what a negative result will look like and what the tube looked like in the beginning. Because most of you guys have blue tubes, right? But in fact, the uninoculated tube was green. Why does it change colors? It's another pH change, right? In this case, an alkaline pH change. What does this tell us about your organism? It tells us if it has citrate permease. This is a protein embedded in its membrane that allows it to take citrate up from the media, right? So this is a different permease. This one's for sucrose, right? But same principle. It can't get in unless it's got a door to get in. So does your organism have that door? Does it have citrate permease? Can it take citrate from that media and bring it into your cells, into their cells? So who's got a blue tube? Hold it high. Be proud of your blue citrate permease positive organism. Okay. It's basically the whole back table, right? The whole back table. And then this table, one winner and one winner. And that's correct. All the rest of you guys, right? It may be looking kind of bluish, but trust me, it's not blue. Compare it to the positive control, right? Yours is negative. Yours does not have citrate permease. So no growth, and we're going to talk about that. So what's the only carbon source in this media? And your organisms have to have carbon to grow. Sodium citrate. That's it. And then they also need nitrogen. We also only gave them one nitrogen source. And this is so that we can indicate what was going on pH-wise as well. The only nitrogen is ammonium dihydrogen phosphate. Because of that, since this media only has one carbon and one nitrogen, what will happen to the organism if it cannot take in these molecules into their cells and use them? It won't survive. It will die. So if you have a non-bright blue tube, right, and you think you're seeing growth on that slant, you're not. What you're seeing might be the fact that you put a lot of bacteria on that slant to begin with. It's what we call a heavy inoculum. At this point, if it is not the same bright blue as that positive control on your lab bench, it is negative for citrate permease. And it is, in fact, dead. It's dead. Well, we're going to talk about it. I haven't gotten to those questions yet, y'all. You're skipping ahead. Okay, what's the pH indicator in the media? It's bromenthal blue. So it, around pH 7, which is about where your media starts out, it's about green. When it goes above, well, wait a minute, at 6.8, excuse me, right below 7, it's green. As it increases in pH, it's going to turn blue, right? So let's know the pH went up, which means that's basic, right? That's less hydrogen ions. And that's because, so it turns blue, right? So decrease in hydrogen ions, increase in pH. They consume the ammonia, right? The ammonia the ammonia gets consumed, and you get the conversion I always screw this one up. It's going this way. Without, I forget. Oh, good news, I'm not asking you specifics. Yeah, it loses hydrogen ions, right? So, right, again, when hydrogen ions decrease, pH goes up. That's the important thing, as it consumes the ammonia. Like the phenol red test, when you have increasing or decreasing hydrogen ions, this means that you have an increasing pH, which is a different color change, right, for phenol red, because that's a different pH indicator. In that case, it turns that pink magenta color, right? If the media is green and you see growth of the organism in the tube, is this a positive result for the utilization of citrate? If you truly see growth, yes, right? If it truly is growth, right? But again, be careful, right? Special note, only record in this class a positive result for your unknown if the tube is blue. If you think you see growth and it's not blue, it's probably a heavy inoculum. Like I said, you put too much bacteria in there to begin with. It didn't actually grow. That's not growth that you're seeing. Okay? Those of you guys that have blue tubes should have good growth, all right? See nice good growth. Lawn growth. Does that make sense to you guys? Okay. So sometimes some scientists will actually stab the butt end down here, with a needle instead of just inoculating the slant. I don't mess with that for you guys because we incubate it long enough that if it's positive, it turns blue. No problem there. So explain how a citrate positive organism might not produce a color change. Well, think about it. Think about your organisms. What they need to grow, right? And they need to use a lot of it to cause this pH change. So what factors would affect its growth? What'd you say? Incomplete incubation, maybe the wrong temperature, right? Someone stuck it at a cooler temperature, it's growing slower. Maybe you didn't have a lot of live cells. Maybe you had mostly spores, right? Improper pH, right? If the media wasn't made properly. Not a problem there, right? So is this a false negative result? Yeah, right? If it's growing, but you don't see a color change yet, right? And this would be sensitivity or specificity issue. Sensitivity, right? Not enough of the substance to create that color change. Alright, so what does this all mean? An organism is or is not capable of fermenting citrate and consuming ammonia as the sole carbon and nitrogen sources respectively. Right? It either has citrate permease enzyme in order to be able to bring the citrate in and grow, right, and then change the pH, or, right, it does not have that ability. Some have additional enzymes like citrase that can break down citrate. So who is yours? Is it is or an is not, right? This is a pretty cool test that we no longer do because each one of these tubes, and I don't even have, we've discontinued this so long ago, and my refrigerator died that I stored them in, so I lost all my real world examples of these. So I always have his pictures in the book and diagrams. But these are called enterotubes, and these would be for enterobacteria, so any of your organisms that fall into that classification. Basically, none of the gram-positives would we be able to test using this. There's another test system for gram-positives. But for the gram-negatives, what we could have done was inoculate the end of the needle that's underneath this cap and inoculate all these chambers all at once, right? So you're running 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14 tests all at once in one fell swoop. Then you come back the next day and you interpret the results. And some of these are similar to what we did, right? Notice that this chamber is a citrate chamber. So it starts out green, and if it's positive, it turns blue. These first 3 chambers have a layer of wax over the media. This is to create an anaerobic environment. For glucose fermentation, we know if gas is produced because it'll push the wax up. Where we were able to detect gas production, right, with the phenol red test because what was in our tubes? Another tube, a little Durham tube that upside down that traps the gas. So here it would push up the wax. And again, same yellow color change for acid because it's phenol red. And guess what these guys are? Adenol, lactose, arabinose, sorbitol. These are all sugars. These are all phenol red agar, and they will turn yellow in the presence of acid if they actually ferment that sugar. We can't tell gas production because there's no wax in these chambers. These other 2 right here are ones we don't perform. These are those decarboxylation that I mentioned earlier, how they can break down some amino acids. Same is true for phenylalanine deamination. In this chamber, it can either do that or it can ferment the deuterosol, another sugar alcohol. You can puncture open this chamber and add the VP reagents, like how we did it in a broth tube. Here you can add it to the surface of the media, and you can get that red color change if it is in fact VP positive. This is a cool test. This is one of Taquana's favorite. It's for urea hydrolysis, and it turns this really pretty magenta pink color if it's positive. She always says that when she gets married, her bridesmaids are going to wear that color. So, you know, I think I'm going to opt out. Just kidding. This one is 2 tests. If hydrogen sulfide reduction, if that happens, if sulfur reduction happens, you get a black precipitate. This one you can also open the chamber and add the Kovacs reagent, and you can test for the idol production and it'll turn red. So you can run a whole bunch of tests. And this is actually a numbering system. And one other professor that I pointed you guys to her videos before, right? She's pretty cool. She runs through the whole thing and tells you all the really cool stuff about the organisms that we would perform this test for. So it's kind of long. Well, it's 18 minutes. It's not bad. She's really good, though, if you're interested in learning about that. In clinical research labs, they use that kind of testing all the time. I wanted to go back to my PowerPoint. So there is nearly 14,000. I'll have to look. I don't know where my books went. There's a book that comes with it, and there's actually a coded system. And you figure out a coded number, and then you look it up in the book, and it'll tell you what organism or what organisms this might be when you get your results. It's really kind of cool. So we already just talked about this in the beginning, right? How you can narrow down your possibilities. So definitely there's some problems with MR or VP testing. I would go by those. I would go by all your other tests first. And don't base your results on MR, VP. So this is an interesting thing that happens. Most bacteria will respire aerobically when oxygen is present, if it can, right? This applies to obligate aerobes, facultative anaerobes, and microaerophiles. They will obtain a small amount of energy via glycolysis, but they will capture a relatively large amount of glucose's free energy from oxidative phosphorylation, using that electron transport chain and having the oxygen accept those final electrons. The main role of the Krebs cycle in energy production is to feed, remember, all those electron carriers, all those electrons from breaking down glucose to the electron transport chain. That's where the bulk of the ATP is produced. They recycle their reduced coenzymes, right? Whenever the enzyme drops off the electrons of the electron transfer chain, it goes back to the Krebs cycle, picks up more electrons. And it just keeps shuttling back and forth, back and forth, back and forth. As long as the system keeps working, as long as oxygen is there and the electrons can be accepted by it at the end of that electron transport chain. Alternatively, they can break down sugars like glucose, right, and all the way to carbon dioxide. Many of them can resort to fermentation when oxygen isn't present. So again, those electron carriers can't go to the electron transport chain because if they go down, boom, they get to the end and there's no oxygen. So instead, those electron carriers, remember, need to regenerate their ability to pick up electrons, right? The electrons have got to go somewhere. So they actually add it back to pyruvate, right? We break down glucose in the process of glycolysis into pyruvate. And then they add those electrons back to different components of pyruvate, right? Different edifications, right? All the different possibilities with fermentation, right? But the take-home message is mainly we produce what? 3 main compounds can be produced from fermentation in general terms. What does the yellow tell us in the phenol red tubes? Acid. What does the bubble tell us? Gas. What does the VP test tell us? Alcohol. Right? But remember, they only get a little bit of energy, ATP, from this, from that substrate level phosphorylation. They're not doing oxidative phosphorylation, no electron transport chain. But that's better than dying, right? So there's our alcohols, acids, and gases. So there's something peculiar going on here. Most bacteria, as we said, will respire aerobically when oxygen is present. Is there oxygen present in this lab? Yeah, I should hope so, otherwise we'd all pass out and die. Many organisms, many bacteria can resort to fermentation even when oxygen is present. Oh, when it's, yeah, they're going to resort to that when it's absent. But under some circumstances, they will prefer to ferment even when oxygen is present. I jumped ahead. Okay? So, when oxygen is present, let's use it. We're gonna make basically $30 an hour, right? When oxygen isn't present, you've got to ferment. You're only going to make $2 an hour. So, why would you choose when oxygen is present to actually ferment and only make $2 an hour as opposed to using aerobic respiration, which would earn you $30 an hour? That's perplexing, is it not? Why on earth would you do that? You have oxygen. Why aren't you aerobic and respirating? Why are you fermenting, right? Look at your tubes. There's oxygen in here, but that's fermentation, is it not? We've got acid, we've got gas. So why do they do that? It's called the Crabtree effect. Of course, named after a scientist. Over short-term periods of time, the preference of glycolysis over aerobic respiration is when conditions of glucose or that sugar is high. Because, as an example, this is not true numbers, right? But as an example, and this is probably what happens to people when they win the lottery, right? They have this huge amount of money in a short period of time, so what do? They spend it like crazy because, you know, they don't know what else to do. So these bacteria, right, if they have, this isn't the numbers. Here, I want to get to the numbers because this really helps. So Peter's the numbers guy, so he put all the numbers together. So suppose you can make 30 ATP for every glucose you metabolize aerobically. But it takes you a second, a tenth of a second, to complete each cycle. So, say per second you generate per glucose molecule 300 ATPs per second. Make sense? Okay. But on the flip side, even though you only get 2 ATPs if you choose fermentation, just glycolysis, right? But it only takes you 0.005 seconds for each cycle. That means that you could complete and make 400 ATP per second doing glycolysis and fermentation as opposed to aerobic respiration, which takes longer. Even though it yields more, because you can do the other one faster, you can actually make more ATP in the same amount of time if you've got plenty of sugar to start with, right? So if you have a ton of sugar, why not just burn it up, right? Who cares? $2, right? But if you have a smaller amount of sugar, you're going to be more conservative with that, right? You're going to take longer. You're going to break it down completely and get your full $30. Right? So, I mean, it's not all that different, right? Money, right? This is the money currency. This is how the cell runs. It's all about energy, right? So, you know, if you pay somebody $2 an hour to do a task and someone else $30 an hour, who's going to really do the good job? Probably the person that's getting $30 an hour, right? The person who's getting 2, he's like, screw you, right? Make sense? They're not as efficient, right? But do you need them to be, is the real question, right? Does this make sense to you guys? Right, so that's why even though, and although in a broth, right, broths tend to get anaerobic, right, because oxygen's not going to diffuse all the way down into that broth unless you're shaking those tubes. But there is enough oxygen there that they could do aerobic respiration. They choose not to because there's a lot of sugar there. So they're like, hey, we're just going to gobble it up. We're just going to ferment it. Right? So if this kind of stuff is really cool to you, like how fast enzymes are, right? Enzyme kinetics and how awesome proteins are. Although some of these numbers I think have changed. But we have a biotech program here at Delgado, and their main lab is actually right across the hall from us. We borrowed stuff from them, right, this semester, like micropipetters. Hopefully soon we'll get our own. But they offer courses, and these numbers may have changed, so check into them. But this course that you're taking right now is a prerequisite to their program. It's one of the requirements for that program. So if you like lab work, you think this kind of stuff is cool, right? The other programs you may be trying to get into don't work out. Check out Delgado's biotech program. So that brings us to the plate media, right, that we're going to look at. So all three of these medias are both selective and differential. So they select for a particular group of organisms by inhibiting a different group. Now, 2 of the plates do that based on gram-positive versus gram-negative. The 3rd plate that we haven't talked about yet isn't based on gram-positive versus gram-negative. Then, if it actually grows, right, if we select for it, then we can ask the next question, and that's a differential question. What can your organism do, right? And how does it differ from others that don't do this? And so this is usually biochemical characteristics, right? And again, the question that we ask a lot in this lab is fermentation, right? Does it ferment a particular sugar? So for mannitol salt agar, mannitol is a sugar in its alcohol form. So yet again, we're going to be asking if the organisms grow, does it ferment this sugar? But we're going to select for a particular group of organisms with this media. What type of organisms are we selecting for? Typically, with clinical specimens, we're looking for Staphylococcus. But remember, we're not dealing with clinical specimens, pathogens in this lab. Not anymore, anyways. So, what makes this selective, though, for Staphylococcus? What's selective about this media? It's salty, right? It's more salty than most media. It has a higher than normal salt concentration. So if your organism grows on these plates, y'all, it isn't necessarily a Staphylococcus. But what you can know definitively, it is salt tolerant, right? For high levels of salt, which of course most Staphylococcus are, and that's why we use this media. But this is important. You need to put high salt concentration, not just salt, right? Most media has salt for isotonic balance. The important thing here is the high salt concentration, right? Higher than normal. Does that make sense to you guys? Okay. And that's how we can normally select for Staphylococcus, but note that some of you guys are growing, but so this is, this is much higher than normal saline, which is around 0.9%. This is 7.5% salt. This is very salty. When these plates start to dry out, you see salt crystals forming. There's so much salt in them. So these guys start out this nice, pretty pink color. Because what compound in this media are they going to actually change? What makes this media differential? So salt is selective, right? What's the other component? Mannitol. What is that? It's a sugar out in its alcohol form, right? What do most organisms do to sugars? Ferment them, right? So mannitol is our differential agent. Without this, we're not going to tell mannitol fermenters from non-mannitol fermenters. So they can break it down, right? And fermentation, as we know in general, they could produce acids, alcohols, and gases. Which is the easiest to detect the production of? An acid, an alcohol, or a gas, especially on plate media. One of these is definitely eliminated. We can't tell if it produced it, right? Yeah. Gas, right? We can't tell if it produced gas in this media, right? It just goes into the air, right? Can we detect alcohols? Not really, but we can detect acids really easily using what? pH indicator. So what's the pH indicator in this media? We've already used this one. Phenol red. Phenol red. What color does the media turn if the organism generates acid byproducts? Yellow. So there's a beautiful streak plate, really beautiful, right, of some Staph aureus. and tucked away in my refrigerator. Shh, don't tell anybody. I had one plate left. So it's taped shut. It really is Staph aureus. But most of us have this in our nose, y'all. So notice my uninoculated side and then the yellow side, right? Because that's Staph aureus. It can grow, right? Salt doesn't affect it. It's salt tolerant. It's growing on that plate. And it's fermenting the mannitol and producing acid, turning it yellow. You guys can pass that around. It's taped shut. It'll be safe. So what's the name of the organism most commonly found in clinical specimens that can grow on this media and in the process generate acid byproducts? Staph aureus. Staph aureus. None of you guys have Staph aureus. So who has a yellow plate? Yeah, hers is growing and it's yellowish. Hers is growing and it's yellowish. Ooh. But I already told you it's not what? It's not Staph aureus, but it is salt tolerant. It is fermenting mannitol. But it's not a pathogen. So we didn't do this. Why would you put the organism on a nutrient agar plate? What would be the purpose of doing that? So you do a salt agar plate, and most of you guys have no growth on these plates, right? Does anybody else think they have growth? Nobody has Staph aureus. Nobody has Staph aureus. There is another staph, Staph epi, that grows on your skin, which is safe. So does anybody else think they actually are seeing growth on their mannitol salt plate? The pink one, yes. Probably not. A little bit. Yeah, a little. Yeah. You might have significant growth. Do you see hers? Right? But her plate's not yellow, right? The media isn't turning yellow. Is yours gram-positive? Oh, that's Latrice? That's it's Amber's. Yours is gram-positive? Did it grow on the blood agar? Okay, yeah. So yours might be Staph epi. Yeah, okay. Mike, do you have growth on the blood agar? Yeah, you do. That's yours, right? Well, that's yours. All right, so what about... Yeah, that's the blood agar. That tells you it's gram-positive. Which side of that plate is hers? Ah. So you might have Staph epi as well. You do. Okay. Yeah, you have Staph epi. All right, so y'all see her plate? And it's actually starting to turn a little yellow. We find this kind of happens with Staph epi if it goes too long. It's not Staph aureus, okay? We're good. So why would we want to do, so most of you guys, it didn't grow, right? Well, does that mean your organism was dead? No, it means the salt killed it. It's not salt tolerant, right? Can we ask the second question about your organism? Can we ask if it ferments mannitol? No, because it died, right? So it's not applicable for most of you guys, right? Mannitol fermentation, we can't tell, right? Two of you guys, right? Probably you've got yellow plates, right? Again, it's not Staph aureus, but it is a fermenter of mannitol. I would say non-fermentation wamba. I wouldn't say that's truly yellow on that one. Okay? And then yours, or Latrice, or whoever it is, Amber, right? It's growth, not yellow, right? Okay. Yes, it's definitely salt tolerant. You definitely have growth. Yeah, yours might be salt tolerant, and actually your plate, do you notice it's kind of that bright pink like your phenol red tubes? And that's because there's protein in this. So again, yours is probably digesting the protein. That's why it's getting pinker. No, not mannitol, protein. Right, doesn't ferment. Non-fermenter. All right. So why would Nutrient Agar help us? Because most of your guys were killed by the salt, right? But this would help confirm, if it grew on the Nutrient Agar, that what killed it? The salt, right? This would be a control. We didn't bother with this because you guys have been doing this all along, right? You have Nutrient Agar slants. You know your organism grows, right? You know it's the salt that killed it. And in clinical labs, they wouldn't do this either, right? They just suggest this in the lab manual, again, as a control so that you guys could see. And we have been doing it. We just didn't do plates. So what would be the likely consequences if we emitted the salt from the mannitol salt? The plates would no longer be what? They'd no longer be selective, right? Anything would grow. And that would be good because part of the reason why we're using that media is to select for salt tolerant, like Staphylococcus. Right? And so you would get a bunch of false positive results because you'd have organisms growing on those plates that aren't supposed to be growing. Would this affect the media's specificity or sensitivity? This would be specificity, right? It would no longer be specific for salt-tolerant organisms. Okay. So remember, organisms need to have carbon and nitrogen. So what gives us carbon and nitrogen in this media? Well, the beef extract, right, has everything. Y'all, it's animal cells. It's cow cells broken down. Everything is there. Carbohydrates, proteins, nucleic acids, lipids, you name it. It's cells, right? So there's no shortage of both carbon and nitrogen. Peptone is a pre-digested protein, so again, lots of carbon and nitrogen. Mannitol is only carbon, right, and water, sugars, or carbohydrates, right? C, carbon, hydrogen, and oxygen. No nitrogen. Know that the energy can be derived from carbohydrates, right? Most organisms we prefer carbohydrates, sugars, right? But in the absence of sugars or in the inability to break down a particular sugar, the organism can get energy from proteins. And that's the case for Anti-Intermera's organism, right? Yours prefers the protein, right? And turns that media alkaline. You get those bright pink colors. And again, sugars yield more energy than proteins do. With the diversity of microorganisms in the world, how can a single test such as the MSA be used to confidently identify Staph aureus? This is more like a true and false, right? Could you? No. Definitely not, right? In this lab, right, you've got organisms that are fermenting the mannitol and growing on these plates, but they're not Staph aureus. I already told you we don't have that in this lab anymore, right? But in a clinical lab setting, right, there's a pretty good indication that it's Staph aureus, but they would do additional testing, right? But this would really narrow it down, the possibilities, quite quickly. So, do you see growth? Yep. What does this tell you about the organism? Salt tolerant. This is actually Staph epi and a student that didn't know how to do streak plate method. You've seen this picture before in black and white. They inoculated each quadrant separately from the original culture. Just shows that repetition is necessary to learn these techniques, right? So, Staph epi, because again, no color change, right? So it just grew, it's salt tolerant, but doesn't change color. What would you see if the plate were Staph aureus? Right, the plate would turn yellow from the fermentation of the mannitol, producing the acid, lowering the pH, causing it to turn yellow. So here's a side-by-side comparison of Staph epi and Staph aureus on this media, right? You can see growth on both sides, but only the Staph aureus turned it yellow. This plate, right, is another organism, and you can see it's kind of orange. So it's starting to ferment the mannitol. And definitely salt tolerant, right? You see all that growth. But it's not it's actually not a Staphylococcus. But it's also not a clinical specimen. It's actually something you get from the soil. So, good place to look for your write-ups. One of the bacillus species, and actually, who's got the yellow plate? What do you think you've got? Bacillus. I won't spoil it for you yet, but. Nope. It's one of the bacillus. We'll get to it. Okay, so the Columbia CNA is going to give you the answer, right? So the CNA stands for colistin and nelidiscic acid, right? These are pre-made plates, and we use these to differentiate hemolysin ability, if they produce hemolysins that break down red blood cells. So these are exotoxins that will lyse red blood cells. Why do they produce this? What element are they after that's in high concentrations in our cells? Remember, elements are things on the periodic table. Carbon, hydrogen, oxygen, iron. They're after iron. They need it for their metabolism too. So for what group of organisms are we selecting for with this plate? Gram-positive. What are the selective agents? The two antibiotics, right? Colistin and nelidiscic acid. Who do they kill? Gram-negative. So only if you have a gram-positive should you see growth on these plates. Doesn't guarantee you'll see growth, but you should. So gram-negatives like E. coli, you should not see growth on these blood agar plates. So notice the similarity here. Colistin, E. coli. This is awful differential, right? So what's the component in the media that lets us know if these organisms can lyse red blood cells? The red blood cells. The intact whole red blood cells. That's why this media is blood red. And as you can see, we purchase it from a manufacturer so it has their stamp on it. We don't mess with blood. Don't pour these plates. So there's two possibilities that you could see, and this is best observed when you hold these plates to the light, and I have controls for these as well. So beta hemolysis, right, which is complete destruction of the red blood cells. When you hold up your plate, you should be able to see through it, like I can see you guys through, right? And if you notice, and I don't expect you guys to memorize this, on your unknown report, you'll notice that Bacillus cereus is beta hemolytic, right? As well as, where's my other blood agar? Oh, there it is. As well as Bacillus subtilis, right? You can see that one's beta. We don't have any alphas, but most people have streptococci in their mouth, streptococci mutans. It is alpha hemolytic. This is actually a swab of my mouth. And when you hold it up to the light, you can see that green as opposed to the clearing, which is beta. So this one's beta, this one's alpha. Bacillus megaterium is gamma. It doesn't lyse the red blood cells and should grow on the plates, but I couldn't get mine to grow either. So If you have a bacillus and it didn't grow, it's probably Bacillus megaterium. Because I did go back over and swab your guys' Bacillus subtilis or cereus. It's actually subtilis we usually have issues with, to make sure that you've got your beta hemolysis that you see here. So you can pass these around and look at them in the light. So who thinks they have beta hemolysis? Right. So what do you think you have? Bacillus cereus or Bacillus subtilis? Cereus, subtilis. Anyone else? You have nothing. Okay. Oh, you don't have bacillus if you're the front tables. Yeah. Nobody at the front tables probably says hemolysis, right? Yeah. Yours would be called gamma. It just grew. It didn't change the red blood cells at all. Yeah, so actually, pass around y'all's plate. So guys, this is a good example of gamma. It just grew, didn't do anything to the red blood cells. So beta, how I always remember this one, I always think B for bad. It completely destroys the red blood cells. Like I said, you can pick up the plate and you can see right through it. Alpha, on the other hand, is incomplete lysis. It pokes holes in the red blood cells, oxygen gets in, and you see that green discolorization. The only plate that shows the green is that one right there, right, with the square or rectangle, excuse me, and that's for my mouth. Your mouth would look the same. So, as I said, hold it up and look at it through the light. So these are examples of alpha. You see how it's greenish? Nobody has alpha. If you had Staph aureus, this one is also beta, which is why it's bad, right? Why it's pathogenic, right? It can completely annihilate your red blood cells, right? So this one's beta. Is it gram-positive or gram-negative? Gram-positive because the colistin and nalidixic acid will kill the gram-negative. So this one's gram-positive. So only if you were gram-positive should you see growth on this plate, right? And if you saw growth, you may see lysis of the red blood cells. You should only be seeing beta or no destruction. Yeah. Did you pass that one around? Oh. So when it doesn't lyse the red blood cells, it's referred to as gamma, right? Which I hate because gamma hemolysis sounds like it's lysing, but it's not, right? Gamma is not lysis. So it just grows, doesn't destroy the red blood cells at all. Because there's other stuff in the media to eat. So this is an interesting plate. This was a really good streak plate that a student did. But unfortunately, she did not hold probably her lid over her plate as she did her streak plate. The reason why I know this is because this right here is beta hemolysis, and that's contamination, right? Her organism is gamma. It's growing just fine everywhere else on the plate. This little spot right here is contamination, probably from her own nose, breathing on her own plate. Because most people have Staph aureus in their nose, right? And on blood agar, it's beta hemolytic. And that's what you're seeing here is beta hemolysis. Here's Staph epi, by the way, on mannitol salt. And notice how the organism will be kind of yellow, but the media doesn't turn yellow. So I forgot I had this one. Go ahead and pass that one. So you see, and then over time it kind of will look like that. So here's a whole bunch of just more examples of hemolysis, right? Here's beta, right? Here's different degrees of beta. That's beta and that's beta. That's alpha, that's gamma. That's gamma. See, it's just growing. Here's gamma next to beta. So many, but not all, species of streptococci are beta hemolytic. So these are the ones like the ones in my mouth are not beta. But there are different tests that are run on the polysaccharides that are associated with their cell walls. And that's the Landsfield classification. Hemolysins produced by some streptococci are actually even called streptolysins. They're given a special name. Some of them are oxygen liable, which means they're destroyed by oxygen, where others are oxygen stable. So an additional test that you can do, but we don't usually do in this lab, is you could actually poke into the agar. So then that would create an anaerobic environment for your organism to grow in. And you can see if you have alpha on the surface, but you see that clearing, they have beta inside the agar. It's kind of hard to see in this picture. So there's alpha, but there's beta, where it's been stabbed in. So serological testing using antibodies can be used to classify out different groups, and so we actually have a lot of different groups. When we do the ELISA test, our last lab, where it's a similar test where we're using antibodies to help detect what molecules are present on a cell. So you can see there's lots of different groups in this Landsfield grouping. The beta strep test, where is that one? I'm trying to remember where it is. So these are two different things, right? You have beta hemolysis versus alpha, and then additionally they'll usually group them based on these polysaccharides in the Landsfield groups. I thought there was something else. There's a streptococci they test for women when you're pregnant that sometimes we'll have in our vagina naturally. It doesn't cause problems for us, but when the baby passes through, they can pick up that bacteria and get a really nasty infection. So they'll give the mom antibiotics before giving birth to help minimize the passage of that to the baby. And they call it the beta strep test. So looking for streptococci that I think are beta hemolytic, but they also might also fall into Landsfield group B. I'm not sure. I thought I had it written down, but I don't. But that's of, importance medically. So early formulations of the Columbia CNA use a slightly higher concentration of the two antibiotics, but it was reduced later to improve the recovery of our gram-negatives. Because remember, anything in too high of a concentration, including water, will kill you. Right? If you don't pee, you will die. So they found that they didn't have it quite adjusted right. Right? So they decreased it. So what is the question here? Why are some reasons that the higher concentration was less effective at growing gram-positives? Well, like we said, anything at too high a concentration is going to affect anything, right? Anything too high a concentration can be toxic. But also it has to do with how these antibiotics work. Colistin deals with membrane integrity, and of course gram-negatives have two membranes, so they're really susceptible to this type of antibiotic, as opposed to gram-positives, which just have the one membrane. And then the other one interferes with DNA replication, and between these organisms they use enzymes that are a little bit different. So this one doesn't usually have too much cross reactivity, right? Pretty much just targets gram-negatives. It doesn't affect the gram-positives. But these are important, right? When we use antibiotics, different ones affect different things, right? Like penicillins affect cell walls, right? Peptidoglycan synthesis. We don't have to worry about toxicity with those because we don't have that, right? The things that mess with membrane integrity and stuff like that too, you won't give a patient colistin, right? It could affect our membranes as well. These are only used in these plates. So this goes into some more specifics, right, about that. We're going to skip over that. That's FYI. For your information, not for your testing. Okay, so the last one is eosin methylene blue. There's two different formulas out there, one that contains sucrose and one that contains sucrose and lactose. Excuse me, there's one that contains just lactose and one that has sucrose and lactose. We use the formula that just contains lactose, right? So our EMB plates only contain lactose, they do not contain sucrose. What group of organisms are we selecting for with this media? Gram-negative. So if you have growth, right, this confirms yours is a gram-negative, right, on these purplish plates. Right? Yeah, because yours is gram-positive. So what's selective? What stops the growth of gram-positives on these plates? What is eosin and methylene blue? They're dyes. What do dyes do to organisms? They'll kill them. The gram-negatives are protected by that outer membrane. Where the gram-positives, this gets in and it kills them. These two dyes, eosin and methylene blue. So who can't grow? The gram-positives. What components make this media differential? What are the organism going to ferment and produce acid byproducts from? Sugar. What sugar? Lactose. Right? You can buy one with lactose and sucrose, but remember for our lab, it's just lactose. Why do scientists use this media? Typically they're looking at coliforms and specifically maybe fecal coliforms. They basically want to know, did poop get in their sample? Right? This is what you're waiting for whenever we have a boil water advisory. For some scientists to plate out stuff and see if it grows on plates like this. Of course, we don't want to see any growth on these plates, right? Because that means that poop got into our drinking water. Coliforms is a broad classification given to organisms that are found in the intestines of warm-blooded mammals like ourselves. They tend to be lactose fermenters, hence why there's lactose in this media. They're gram-negative bacilli, and they produce acid and gas from fermentation of lactose. Fecal coliforms reside in the intestines, right? And so therefore found in fecal matter. And therefore if you find them in a sample, it probably indicates that that sample has been contaminated with feces. The coliforms themselves, like E. coli, aren't necessarily bad, right? Most E. coli are non-pathogenic and we all have got it. The problem is if E. coli is there, that means poop is there, y'all. That's where E. coli comes from. So that means other bad things like salmonella, shigella may also be there. Those guys are gram-negative, but they're not lactose fermenters. You wouldn't see them differentiate lactose fermentation on these plates. So these guys will grow right under these specifications. And again, the presence of them, say, in our drinking water is not a good thing. It means that other organisms may potentially be there that could cause disease. Approximately 90% of fecal coliforms are E. coli. Other genera that are present are Klebsiella and Citrobacter. It's often these other genera in the fecal contamination are pathogenic. When the pressure drops in New Orleans water purification plants to 15 psi per square inch or below, the city issues a precautionary boil water order for 24 hours while it checks for coliform contamination of our drinking supply. What's the good news? Every time they've done that, the results have come back negative. We didn't actually have contamination. Right? So they would filter the water sample, trap the organisms onto a membrane, put them onto the media, and then they would look for growth the next day, right? This is why we have to wait for results, to wait to see if anything grows. Right? If you see something like this is McConkey agar. It's similar in that it can detect pH, and this is growth. And this is a lot of growth on that membrane. So you would not want to drink that water. Right? Potable water, drinkable water, has less than 1 coliform pore per 100 milliliters of sample tested. So if there's like 1 in there, it's okay. If there's more than that, then we lose our minds. So these are all the times that we've been told to boil our water. And thankfully every time it was just that, precautionary. There was no actual contamination. One of these I didn't even know about until after it was over. I was like, oh, great. Foods can be contaminated from a variety of sources, right? Our soil, our water, enteric microbes of humans and animals can have several different genuses that can be a problem. C stands for coliforms, right? So these are ones found in our gut. They also can be found in the environment, right? So notice some of them in the soil, in the water. So the one that we really exclusively is just found in our intestines is E. coli. So again, that's a strong indicator. And that's also why I prefer to use the EMB plates as opposed to the McConkey. Is that what does E. coli look like on these plates, y'all? That metallic green sheen, right? It's very distinctive on these plates. Yeah. So these are the bad guys, right? Shigella, Salmonella, right? These would be ones found in feces potentially, right? So if we have E. coli there, or even Klebsiella pneumoniae, right, it would be possible that these other guys are also present. There is a pathogenic strain of E. coli known as E. coli 0157. It has acquired the Shiga toxin from Shigella. So it's quite horrible strain. And if you had it, you would be in class right now. It's that bad. So how do foods become contaminated? Unsanitary slaughter and processing practices. I didn't pick that picture. Water and soil contamination, followed by crop contamination. Direct contamination by food handlers and preparers. Right, so my students sometimes say to me, it's like, I can't believe you still go out to eat at restaurants. And I said, well, yeah, I risk it, because I hate to cook and I don't like to do dishes. But I'm also very choosy as to where I go. This is also why, right, every single restaurant that you go into, in the restaurant, somewhere in the restroom or near the restroom, it says all employees must wash hands before